Good morning, and first of all, I'm sorry for my terrible English, but anyway. Probably you know better than me what Gcmaf is. GC protein-derived macrophage activating factor. And you know that GC protein is another name for vitamin D binding protein. So Gcmaf belongs to the vitamin D axis elements. Here we have the first report mentioning Gcmaf from Yamamoto in 1994. And here we have the structure of the GC protein with the three sugars, important sugars that are linked to the protein in a branched structure. So in this case, you can observe that it's not important if in synthesized Gcmaf we use beta-galactosidase or sialidase. But probably there are other hypotheses on different structure of the GC protein. For example, this linear structure of the sugars. And in this case, it's critical if using first sialidase and then beta-galactosidase. And this detailed biochemical knowledge of the arrangement of the three sugars in the GC protein molecule is fundamental in designing the laboratory procedure to synthesize fully active Gcmaf. And this information is somehow encrypted, and it took all the professional expertise of immunobiotech to synthesize a fully active Gcmaf molecule. So we have to consider also this possibility. A little bit of history of Gcmaf. This is the first report mentioning of Gcmaf by Professor Yamamoto. In 1999, Yamamoto is the first report mentioning the use of Gcmaf on tumor mice. Here in 2008 appeared three reports of Professor Yamamoto mentioning the use of Gcmaf on breast, colorectal and prostate human cancer. In 2009, first report mentioning the use of Gcmaf on HIV-infected patients, and since 2010 we can find in the literature a lot of reports on the application and or role of Gcmaf in other diseases like chronic fatigue syndrome, autism, Lyme disease, endometriosis, and other. Our research group in Florence since 1996 has been researching on the vitamin D axis components. I mean vitamin D receptor and vitamin D binding protein. And since 2009 we started research on the Gcmaf. First, obviously, we study the effect of Gcmaf on immunosystem-related cells, such as monocytes, human monocytes, macrophages cells, and human peripheral blood mononuclear cells. Then we thought also on effects of Gcmaf on different kinds of cells. So we studied the effects of Gcmaf on human breast epithelial cells, human breast cancer cells, and also on angiogenesis. And in these last 4 years, we have some important presentation on national and international congress, and 3 papers published in important scientific journals that you can find on the National Cancer Library. Recently, we focused our attention on human cortical neurons. As Professor Ruggero has mentioned earlier, Gcmaf has many more effects than simply stimulating macrophages. In the following slides, I shall show you the results that we have obtained stimulating human neurons in culture with Gcmaf. This cell line is often used as an in vitro model of neural function and differentiation. The immortalized neural cell line maintaining, it's important, the normal features of neuronal cells. You can see that some of the cells grow into clumps which float in the media, while others form clumps which stick to the dish. So it's not so easy to work with them. We, in particular, focused our attention on the effects of Gcmaf about cell viability, second messenger formation, in particular at the CMP level, cell morphology, and apoptosis. We perform a cell viability assay that is an assay, colorimetric assay for the determination of the number of viable cells, and it utilizes a tetrazolium salt that produces a formazan yellow dye upon reduction by dehydrogenases in the cells. The formation of the dyes increases with the cellular metabolic activity due to elevated NADH flux. So it is related to the mitochondrial activity. Here are the results of the MTT assay. You can see that after a 24 hours treatment at a different concentration, Gcmaf induces a dose-dependent increase of the cell metabolic activity. This means that the molecule is biologically active. On the contrary, its precursor, GC proteins, at the same concentration does not affect the metabolic activity compared to the control-untreated cells. The increase in metabolic activity in response to Gcmaf reflects an increase in the energy produced by the mitochondria. These results might be of great importance in those diseases such as chronic fatigue syndrome or other neurodegenerative diseases where a deficit of cellular energy is observed. We focus our attention on the concentration of 0.4 and 4 nanograms per milliliters. We evaluated that probably they are best concentration for our model. So in the next investigation, we evaluate these 2 concentrations. Here, the second messenger formation CMP level assay, it is a direct immunoassay colorimetric kit. Here are the results, and these results reflecting the Gcmaf-induced metabolic activity, that we can say that they correlate with CMP formation in a dose-dependent manner, and at the same time, GC protein at the same concentration induces a specific CMP increase. So these results are quite superimposable to the previous MTT results, cell viability results. The increase in cyclic AMP appears to be a constant trait of Gcmaf intracellular signaling. In fact, in the past we had observed such as an increase in cyclic AMP following Gcmaf stimulation, also in human circulating monocytes. Here, this response was dependent on the vitamin D receptor gene polymorphism. In human neurons, we don't have results in this sense yet. However, it should be noticed that these neurons show this VDR genotype that is one of the most responsive genotypes. Further studies will assess the responsiveness of the other possible genotypes. Here, the cell morphology. We perform an hematoxylin and eosin histochemical staining that colors nuclei of cells blue and other eosinophilic structures in various shades of red and pink, for example, the cytoplasm. We focused our attention on cell shape and size, staining affinity, and chromatin. Here are the results. In the control cells, the cells appear polygonal with the cytoplasmic elongation arising from the cell body. The mean size of these cells is about 10 micrometers. The cytoplasm is small and highly stained, and chromatin is condensed. In the Gcmaf treated cells, we can see that numerous cells appear roundish without cytoplasmic elongation. The mean size is significantly increased. The cytoplasm is wide and poorly stained. We thought that these are newly formed cells. We have to observe that this kind of cells are present also in the control cells, but the difference is that in Gcmaf treated cells, their number of these big cells increases their number. Here there is an histogram of the number of large and roundish cells. You can see that the number of these newly formed cells significantly increases when treated with Gcmaf in comparison with control untreated cells. So these morphological changes that we observed in response to Gcmaf stimulation, together with the previous increase in metabolic activity that we saw, can be interpreted as if neurons were dividing and entering the process of differentiation. It is well assessed that neurogenesis is in the adult is a fundamental requisite for learning, memory formation, and all superior neurological activities. Therefore, these observations of the direct effect of Gcmaf on human neurons support its use in a number of neurodegenerative diseases, where the number and the arrangement of neurons is affected, from chronic fatigue syndrome, autism, Alzheimer's disease, and to brain aging. We are confident also that the transcranial ultrasonography technique described by Professor Gero before will confirm in vivo these results obtained in vitro with our neurons. And this is the apoptosis. We use a Cas3 active human ELISA kit to detect and quantify the level of human active Cas3 protein that is the major executioner protease in apoptosis. The results. You can see that the blue is after 4 hours of treatment and 24 hours of treatment, the red, and after both 4 and 40 hours, you can see that no significant apoptosis is induced by Gcmaf or also GC protein treatment. And this results is very important This absence of apoptosis in response to Gcmaf is extremely important, possibly even more important than the positive effects so far described. These results demonstrate that Gcmaf does not induce unwanted side effects in human neurons. This point, previously assessed also in autistic children by Dr. Bradstreet, assumes great importance when considering the administration of Gcmaf in the context of the so-called compassionate treatment of patients with advanced or incurable disease. In fact, in many countries, the absence of side effects is a prerequisite for the compassionate administration of substances.